Bioinformatics. Bioinformatics is a discipline that allows applying computer tools to the study and management of biology data. It is also the comparison of data between biological species. The most important thing about bioinformatics is information technology and biology. Biotechnology helps to redesign and modify organisms to do what we want and uses bioinformatics because it gives a very general idea of how genes work and how to predict the functionality of genes. Bioinformatics is based on the comparison of data between biological species. Bioinformatics is a multidisciplinary science that takes the most important concepts from molecular biology, general biology, biochemistry, genetics, mathematics, statistics, and system engineering. It is based on computer programming, that is the bioinformatic software development, the sequencing, that is the knowledge of chemical qualities of nucleic acids and molecular techniques. Coding is an instruction or order. Operative system is the software that works as hardware intermediary, and the script is a set of orders saved in a file.
DNA sequencing is the process of determining the precise order of nucleotides within a DNA molecule. It includes any method or technology that is used to determine the order of the 4 bases: adenine, guanine, cytosine, and thymine in a strand of DNA. The first DNA sequences were obtained in the early 1970s by academic researchers using laborious methods based on two-dimensional chromatography. The sequence tells scientists the kind of genetic information that is transported in a specific DNA segment. For example, scientists can use sequence information to determine which sections of DNA contain genes and which sections carry regulatory instructions, which activate or deactivate genes. In addition, and in a very important way, stream data can highlight changes in a gene that can cause them. New generation technologies allow for cheaper and more efficient sequencing, despite obtaining smaller fragments. These accelerated sequences obtained require the use of powerful computer tools for their assembly. An important point in bioinformatics and computational biology projects is the use of mathematical tools to extract useful information from data produced by high productivity biological techniques such as genome sequencing.
Sequencing is referred as the determination of the exact order of the base pairs of a specific region of DNA. This amplifies the genetic information of the organism that is being analyzed and allows to confirm its identity. Sequencing technologies that have been used during the last few years allow the production of millions of DNA sequences at an unprecedented speed with an increasingly reduced cost per nucleotide. For those isolated sequences obtained, it is needed the help of informatic tools for its correct assembly. Nowadays, the new generation systems available on the market for the use are Method 454 from Roche, Solexa by Illumina, and Solid by Applied Biosystems. For example, Solexa consists of 2 parts. First, the genome is fraction, followed by the clonal amplification of DNA fragments above the solid surface of a flow cell. This way, clusters of clone fragments are created. Then there's a synthesis sequencing, marking the 4 nucleotides by fluorescence of various colors. These modified nucleotides contain reversible endings, which allows that every cycle of the process occurs in the presence of the 4 nucleotides simultaneously. The only problem with this one is that it produces small lectures and it can't resolve short repetitions. With these technologies, scientists can create new applications and biological tests that will change the way pre- and postnatal diagnosis of genetic disorders work. Other types of programs are being developed, hoping they will be faster and give us more information, creating a whole new revolution in genomics. As we already know, DNA contains 4 groups that are arranged in a specific order, which contains genetic instructions to keep an organism alive and function a certain way. To copy a digital file, binary digits represent information on a hard disk and must be translated to the language of the computer. A standard DNA synthesis machine produces the according sequence. These are multiple copies of overlapping fragments, and each fragment contains indexing details that identify where the sequence is stored. This generates redundancy in the system, which means that if some fragments are damaged, the data won't be lost. Also, the same equipment that is used in molecular biology labs to read the DNA of organisms is used to extract the information, which can be accessed on a computer screen. The BLAST family of programs is the most used to look for similarities on a database given a problem sequence. This practice consists of two parts. First, we need to familiarize with the server NCBI, and then we use BLAST to solve the problems. BLAST is a computer program for local sequence alignment, whether we are talking about DNA, RNA, or a protein. This program is able to compare a problem sequence against a large number of sequences that are in a database. The algorithm finds the sequences of the database that have the closest resemblance to the problem sequence. This program, however, uses a heuristic algorithm, so it can't guarantee that it will find the correct solution. But this program can calculate the significance of its results to provide us a reliance parameter.
System packages applications in which it is not necessary to program to relate databases. Firstly, we have Genovis, that has been designed to be an easy-to-use and flexible desktop software application for the organization and analysis of biological data, with a focus on molecular sequences for related data types. For individual use, the price is $395 per year or $1,000 perpetual license. Then we have CLS Genomics Workbench, that is a powerful solution to analyze next-generation sequencing data. Its cutting-edge technology incorporates unique features and algorithms that are widely used by scientific associated with data analysis. Finally, GeneCodes is an international software firm specializing in bioinformatics software for DNA sequence analysis. They make general analysis, Sanger sequencing, and next-generation sequencing. These tools are very expensive. Web Tools for Research, Nucleotides, NCVI, is a series of databases relevant to biotechnology and biomedicine and is an important resource for bioinformatics tools and services. Some of the most relevant databases are GenBank. NCVI has had responsibility for making available the GenBank DNA sequence database, provides gene online Mendelian inheritance in man, the molecular modeling database, a map of the human genome, and a taxonomy browser. The NCBI assigns the taxonomy ID number to each species of organism. Bookshelf is a collection of freely online versions of selected books. BLAST is an algorithm used for calculating sequence similarity between biological sequence such as a nucleotide sequence of DNA and amino acid sequences of proteins. Gene, to characterize and organize the information about genes. A unique gene ID is assigned to each gene record that can be followed through revision cycles. Protein. Protein records are present in different formats, including FASTA and XML, and are linked to other NCVI resources. Protein provides the relevant data to the user, such as genes, DNA, RNA sequences, and biological pathways. Another tool is the DBG Center that is officially certified to collect nucleotide sequences from researchers and to use the internationally recognized accession number to data submitters. Finally, EMBL-EVI that shares data from life science experiments, performs basic research in biology, and offers an extensive user training program supporting researchers in academia and industry. Proteins. UniProt gives protein sequence and functional information. PFAM, the general purpose is
to provide a complete and accurate classification of protein families and domains. It also allows users to submit protein or DNA sequences to search for match to families in the database.
Primer synthesis is the chemical synthesis of relatively short fragments of nucleic acid with defined chemical structure. To obtain the desired primer, the building blocks are sequentially coupled to the growing oligo chain in the order required by the sequence of the product. Upon the completion of the chain, the product is released from the solid phase to solution, deprotonated, and collected. The best programs to work with primers are IDT and SnapGenes. In bioinformatics, a sequence alignment is a way of arranging the sequences of DNA, RNA, or protein to identify regions of similarity that may be a consequence of functional, structural, or evolutionary relationships between the sequences. Global alignments, which attempt to align every residue in every sequence, are most useful when the sequences in the query set are similar and of roughly equal size. This does not mean global alignments cannot start and or end in gaps. A general global alignment technique is the Needleman-Wunsch algorithm, which is based on the dynamic programming. Local alignments are more useful for dissimilar sequences that are suspected to contain regions of similarity or similar sequence motifs within their larger sequence context. The Smith-Waterman algorithm is a general local alignment method also used on dynamic programming. A protein domain is a conserved part of a given protein sequence and tertiary structure that can evolve, function, and exist independently from the rest of the protein chain. One domain may appear in a variety of different proteins. Molecular evolution uses domains as building blocks, and these may be recombined in different arrangements to create proteins with different functions. A protein motif is a supersecondary structure which also appears in a variety of other molecules. Motifs do not allow us to predict the biological functions. They are found in proteins and enzymes with similar functions. In proteins, a structural motif describes the connectivity between secondary structure elements. An individual motif usually consists of only a few elements. In addition to secondary structural elements, protein structural motifs often include loops of variable length and unspecified structure. The best programs to work with domains and motifs are UniProt and ProCite.
How to mark a molecule. A tracer is a substance that reveals its presence in one way or another and can therefore be localized during the course of an experiment. Depending on the substance and the type of experiment, a tracer can be labeled with fluorescence, spin, density, or radiation. In each case, a label group allows a detecting molecule without affecting the specificity of its interactions. For example, radioactive molecules participate in the same reactions as non-radioactive ones, but their location can be followed and it's possible to measure the amount present. Nowadays, there exist three main different ways to mark a molecule. The first one is a radionuclide, which is an atom that has excess nuclear energy, making it unstable. This excess energy can be either emitted from the nucleus as gamma radiation, or create and emit from the nucleus a new particle, or transfer this excess energy to one of its electrons. The second way is by using a chromophore, which is the region in the molecule where the energy difference between two separate molecular orbitals falls within the range of the visible spectrum. Visible light that hits the chromophore can thus be absorbed by exciting an electron from its ground state into an excited state. Lastly, there exists fluorophones, and is a fluorescent chemical compound that can emit light upon light excitation. Sometimes this one is used as a tracer in fluids, as a dye for staining of certain structure, as a substrate on enzymes, or as a probe or indicator. Nowadays, the investigations that scientists can make thanks to bioinformatics have grown for the past years. Here are some samples of the studies that are possible due to bioinformatics. Analysis of gene expression, analysis of protein expression, analysis of cellular organization, Analysis of mutations in cancer. Genome-wide association studies.